Hello students. So today let us discuss the first session of the first unit Mycology, that is the general characters and thallus organization of fungi. This is Dr. Rekha working as Associate Professor in Government Science College, Bangalore. So the study of fungi is called as mycology. So mycology, the word mycology is derived from two Greek words, mycos meaning mushroom, logos means the study. So literally mycology means the study of mushrooms. So one who specializes in mycology, they are called as mycologists. So what are fungi? Fungi are eukaryotic, non-vascular, achlorophyllous, heterotrophic thallophytes. So they belong to a group of thallophytes which is not differentiated into root, stem and leaves. They are eukaryotic because they have a definite nucleus. They are achlorophyllous because they lack the chlorophyll pigment, hence they are unable to synthesize their own food and are therefore heterotrophic in mode of nutrition. There are about 5,100 genera and more than 50,000 species of fungi which are known today. So the common fungi are the mushrooms, molds, puffballs, mildews, rusts, smuts, etc. So coming to the habit and habitat of fungi. So the forms or the habit of fungi, they are diverse. So they range from the unicellular microscopic forms like yeasts to the macroscopic fleshy mushrooms and also the polyporous, which is called as the bracket fungi, which grow as saprophytes on the dead logs of wood and tree trunks. So the habitats of fungi are also extremely diverse. They are ubiquitous in distribution. That means it is seen everywhere. It is found in the soil, rocks, water, air, dead plant and animal bodies, and also on the living plants and animals. So they are also seen growing on the foodstuffs such as bread, pickles, jams, fruits, leather, clothes, timber, glass, paint, plastics, chemicals, etc. Some of the fungi, they grow on the animal dungs and these fungi are called as coprophilous fungi. So coming to the thallus or the vegetative structure. So it may be unicellular or it may be multicellular. Unicellular means it is made up of a single cell like that of yeast, or it may be made up of a number of cells, and it may be filamentous and branched, and they may have a cross wall to divide the filament, which we call it as septate, or it may be without the cross walls, then we call it as aseptate. So the plant body or the thallus or the vegetative body is naked sometimes, and in majority of the fungi the vegetative body is composed of long thin filaments which are called as hyphae. So collectively the hyphae form the mycelium. So it may be, as we discussed earlier, it may be provided with the cross walls or without the cross walls. So if the cross walls, which are called as septa, is present, then we call it as septate, and without the septa we call it as aseptate. So in case of septate hyphae, the cell may enclose a single nucleus, two nuclei or many nuclei, and the septal pores are present, which is called as the dolipore or simple pore. So the hyphae may be eukaryotic or heterokaryotic or haploid or diploid. So coming to the structure, cell structure. So usually when it is examined under the microscope, each hypha, it looks transparent tube-like structure. The composition of the cell wall is variable. So mainly the cell wall is made up of the fungus cellulose, which is called as the chitin, followed by the plasma membrane enclosing the cytoplasm. The cytoplasm encloses the endoplasmic reticulum, mitochondria, Golgi bodies, and the reserve food materials. So you have a definite nucleus, hence it is called as eukaryotic. In higher fungi, we have two groups of fungi, lower fungi and higher fungi. And in higher fungi, the motile cells are absent, but in lower fungi, the reproductive cells are motile with the presence of one or two flagella. The flagella may be whiplash type or it may be tinsel, a feather-like structure. In most of the fungal hyphae, it forms a felt-like structure, and in advanced fungi, you see certain modifications to undergo certain functions, and they form certain specialized structures which we call it as tissues. So there are different types of tissues, namely the Prosynchyma, Plectenchyma, Rhizomorph, Sclerosium, Aprasorium, Historia, hyphal traps, etc. So first let us study the Prosynchyma. So Prosynchyma and Pseudoparenchyma, these are the two types of Plectenchyma. So what is Prosynchyma? So it is a loosely interwoven hyphae and which lies almost parallel to each other, and these cells and the hyphae, they are clearly distinguishable. And second one is pseudoparenchyma. Pseudo, as the name itself indicates, it resembles the parenchyma cells. So this type of tissue is formed by a short-celled and closely packed hyphae. So in this type of fungal tissue, the hyphae have lost their individuality and appear more or less as oval shape or isodiametric shape when you take the section, resembling the parenchyma of the higher plants. Next, third one is the rhizomorph. Rhizomorph in many of the higher fungi, especially the basidiomycetes fungi, some parallel unbranched hyphae, they become closely fused together and grow as a single root-like, rhizome means root, root-like strand. These elongated root-like strands are called as rhizomorphs. And they are blackish and hard on the outside, and they are often found between the wood and bark of the fallen trees or the stumps. So next one is sclerotia, sclerosium or sclerotia. These are more or less rounded or cushion shaped resting bodies formed of pseudoparenchyma. Each sclerotium, it consists of a central medulla of Prosynchyma cells stocked with food, a narrow cortex of pseudoparenchyma cells and an external rind of pigmented hyphae. The sclerotia, they are highly resistant bodies and they are able to survive unfavorable conditions of drought and cold, and later they germinate directly to form a new mycelia, or they may give rise to an external fruiting structure depending upon the species. So next one is Aprasorium. Aprasorium is a terminal, simple or lobed structure which infects the hyphae found in a number of parasitic fungi. It adheres to the surface of the host and helps in the penetration of the infecting hyphae. Next one is the Historia. These are intercellular absorbing structures meant for absorbing food materials from the host. They vary in shape. They may be knob-like, elongated, finger-like or branched. They secrete certain enzymes which hydrolyze the protein and carbohydrates of the host cell. The hyphal traps or the snares. So these are seen in the predaceous fungi, and they form a network of hyphal loops known as the hyphal traps, which helps in capturing especially the nematodes. Next one is stomata. Stomata are thick pad-like solid bodies in or on which the reproductive structures or the fruiting bodies are formed. So coming to the overall nutrition of the fungi. So they are heterotrophic and they may live either as saprophytes, parasites or symbionts. Parasites are those organisms which obtain food from other living plants or animals. Example is Puccinia, Eustallago and Spasilotheca. They may be obligate parasites or facultative parasites. Saprophytes are those which obtain food from dead and decaying organic matter. Example Mucar and Pythium. And these saprophytes may be obligate saprophytes or facultative saprophytes. The third group of fungi is symbionts, where the fungi grow with other living organisms wherein both are mutually benefited with one another. The best example is lichens and mycorrhiza. Thank you.